This slide shows us a drawing that helps us understand the size differential between a bacterial cell and a bacterial virus or a bacteriophage. What you're looking at is a small sliver of the membrane and cell wall that surrounds a gram-negative bacterial cell. How do I know it's gram-negative? Well, I can see two membranes here, and in between those two membranes, I can see a nice thin layer of peptidoglycan. Remember, gram-negative bacteria have a thin layer of peptidoglycan, and gram-positive bacteria have a thick layer. Gram-negative bacteria also have a second membrane. They have the membrane that surrounds the cell, and then they have what's called an outer membrane. Both are membranes, both are a bilayer of phospholipid and protein. But they're going to have different compositions. In other words, the protein that is found in the cell membrane is going to be different from the proteins that are found in the outer membrane. The outer membrane is also going to have a molecule in it called LPS, lipopolysaccharide. In this drawing, the very inside of the cell would be down here, the cytoplasm and so on, and the environment outside of the cell would be over here. I like this drawing. The artist has chosen to add in some additional structures to help us, again, consider their relative size. For example, this machine-looking structure right here looks like a big gear or something inside of a machine. This is a flagellum, or at least a piece of a flagellum. Notice how it's anchored in the cell membrane and again in the outer membrane. The tail structure then extends out into the environment. That's a flagellum. This structure right here, which is also anchored in each membrane, this is a pilus. There are proteins again represented like this one labeled iron transport protein, and the LPS is labeled for us, but this is what we're really interested in right here. This is a bacteriophage. This one happens to be called T1. This one over here is a T4 bacteriophage. Notice there's a little bit of structural difference. The protein in the capsid of the head, this would be a different protein than what would be found in the head of T4. The neck region is also longer in T4 compared to T1. Those are details you don't need to learn, but understand that there are different bacteriophages. So even though they're all similar in shape, they all have that same complex shape, there are different types of viruses that infect bacteria. Notice the size. This T1 bacteriophage is very, very small compared to what would be the entirety of this gram-negative bacterial cell. Now, when we talk about viruses, we talk about them according to these larger groups that they belong in, and we give each one of the individual viruses their own name. Again, it helps us think about viruses in terms of their evolutionary history on the planet, but because they're not living things, they don't get a genus and a species like every other living thing on the planet. Viruses have been named in a number of different ways, and sometimes it gets very confusing because from each type of virus to the next type of virus, the naming system can be very different. For example, some viruses have been named according to the disease that they cause. Think about the measles virus, or the influenza virus, or the rabies virus. Some viruses are named by the geographic region where they come from or where they were first discovered. For example, there are different types of Ebola virus, and each one is named by the region in Africa where it was first observed. For example, Ebola Zaire. Some viruses got named according to the scientist who discovered them. The Epstein-Barr virus is an example of that. Some viruses are named according to their shape. The coronaviruses, for example, got their name because underneath the electron microscope, they look something like a circular structure, a spherical structure, wearing a crown of spike proteins. Someone looked at the coronavirus and thought of those spike proteins like a crown. The term corona translates as crown. And finally, there are viruses that are named by a combination of things. The pandemic coronavirus is formally named SARS-CoV-2, and that stands for Severe Acute Respiratory Syndrome, Coronavirus 2. So you see all kinds of different systems used to name viruses, and it's problematic. It's hard to know, for example, how certain viruses relate to each other or if they don't relate to each other because of this naming system or a lack of a naming system. Now, one thing that has happened in recent years is that the large groups of scientists, for example, at the World Health Organization, have been trying to discourage people from naming viruses after geographic areas. The reason they're doing this is because when a region in a country gets a virus named after it, it often allows other regions and other countries to discriminate against them. Think, for example, about what's been happening around the SARS-CoV-2 virus. People have referred to it as the Wuhan virus or the China virus, and it has even led to attacks against people who come from Asia, as though somehow it's their fault that the pandemic occurred. So very recently, efforts have been put into place to stop naming the different mutations or variants of the coronavirus according to where they emerged. You've probably heard, for example, of the UK strain, or the Brazil variant, or the South Africa, or the Indian variants of coronavirus. Well, we've decided as a collective public health community to stop doing that. Because again, it can encourage some people to just be hateful or act in a prejudiced manner against the region where the virus first emerged. So instead, a decision has been made to name each of the emerging mutations of the coronavirus using Greek letters. So the UK strain, the Brazil strain, the South African strain, all of them are now being renamed with things like alpha, beta, gamma, zeta, and so on. I want to take just a few minutes and talk about the influenza viruses because of all of the different types of viruses, the influenza viruses have the most complex naming system. Influenza viruses fall into one of four groups, what we call A, B, C, and D. Now, the reason that they are put into these four groups is because different types of influenza have different antigens on them. Now, all an antigen is a molecule that is capable of stimulating an immune response in the host organism. Now, the A and B types of influenza are the types that infect a whole variety of animals. The A type of influenza is the type that causes human influenza. It's the cause of the seasonal flu, the seasonal epidemics of influenza that occur here and around the world. The C type of influenza can also cause human disease, but it's very mild. And finally, the D type of influenza only infects cattle, so it would not affect human hosts. So let's take a look at the influenza A viruses. These are the causes, again, of the outbreaks of influenza we see every winter. And in fact, these are also the types of influenza that have caused pandemics throughout the years. You've probably heard influenza referred to with two initials, H and N. For example, back in 2009, we had an H1N1 influenza that caused a pandemic. And a pandemic is simply a global epidemic. Now, lucky for us, the H1N1 pandemic was not nearly as severe as the coronavirus pandemic. The H1N1 strain of influenza just wasn't that virulent. It didn't cause very serious disease. So let's think about the H and N designation. That H stands for hemagglutinin, and the N stands for neuraminidase. Hemagglutinin and neuraminidase are simply proteins, and there are two that are on the surface of the influenza virus. Remember, the influenza virus is a spherical virus. It's enveloped, and it has spike proteins coming off the surface. Those proteins fall into these two groups, hemagglutinin and neuraminidase. It turns out there are 18 different types of hemagglutinin, and they number them H1 to H18. There are 11 different types of neuraminases, so those get N1 to N11. The hemagglutinin protein on the surface of the influenza virus is what binds to the receptors on the host cells and allows the virus to get in to the host cells. The neuraminidase protein is what's going to help the newly made viral particles get back out of those host cells. So the H and the N name that we give to influenza relates to those two very important proteins. Remember, influenza type B can also cause disease in humans, but we don't give influenza B the H1N1 naming system. There are different strains of influenza B, but it just doesn't cause the kind of disease that we see with influenza A, so it's named much differently. Back in 1980, the World Health Organization came up with a system for naming influenza that was shared with the entire globe. And this is the system that we all use now. It's a very complex naming system, though, and it can be very confusing to people. So when you name an influenza virus, first you name its antigenic type. Remember, that's the ABC. Then you name the host of origin. Many influenza viruses first emerge from animals. They come out of swine or horses, that's equine, or chickens or other types of birds. Now, if the host of origin is a human, we just skip this part. In other words, if it's something other than a human, we're going to include the name. The third part of the name is the geographic region where it was first emerging. The fourth part of the name is a number given to the variant or the strain, the specific mutation, in other words. The fifth part of the name is the year that it was first isolated. And then finally, again, for influenza A viruses, we give it an H and an N number. Very, very complex naming system. Now, why do we do this? Why do we come up with such a complicated naming system? Well, influenza is a type of virus that mutates very, very frequently. It mutates each and every year. And so there are constantly new variants emerging, and some of those variants can cause significant disease. As you know, we immunize people against influenza, and people go for those vaccinations once a year, typically in the early winter or late fall months, typically around October and November each year. And what happens is the World Health Organization and other public health experts basically pay attention or do surveillance of influenza outbreaks around the world, and then keep track of which strains are causing disease the most in any one year. And of course, we benefit from that because influenza season, if you will, occurs first in Asia and then comes to the West. So their time of having influenza comes before our time of having influenza each year. So whatever the prevailing strains are in Asia in any one year, groups like the WHO will share that information with each country. And in our country, the FDA convenes a committee each year that decides which strains are going to be most problematic for us. That's how the vaccine is made every year. Each year, we produce 2 different vaccines, typically what's called a trivalent vaccine that has the top 3 strains in it, and sometimes a quadrivalent vaccine. It has the top 4 strains of virus in it. Those strains are mixed up and they become that year's flu shot. For example, the trivalent influenza vaccine that was distributed in our country to help protect us against influenza back in the 2019-2020 flu season contained these 3 strains of influenza. A. Brisbane, O2, 2018, H1N1, and then a strain name. Remember, that's the type of influenza, that's the geographic region, that's the strain number, that's the year it first emerged, there's the H1N1 designation, and there's any additional strain information associated with it. Here's one that emerged out of Kansas, and it was strain number 14, and it first emerged in 2017, and it has an H3N2 pattern of proteins on its surface. This was a B type of influenza that emerged in Colorado. It was the 6th strain or the 6th mutation. It emerged in 2017. These names are very complex, but they make a lot of sense. They allow us to keep track of the many, many different mutations or variants of influenza that emerge around the world each and every year. Now, there's another kind of classification system that I want us to be familiar with, and this one does get applied sort of universally across all different types of viruses. This is not a naming system. This is a classification system. So this doesn't change what a virus gets named as, but this is how we take a virus and we classify it. We put it in with all the other viruses and group it where it belongs. This is the one system that we use scientifically when it comes to classifying viruses. And it's referred to as the Baltimore system. The Baltimore system takes viruses and classifies them according to 4 different things. Number 1, the type of genome that they have. Remember, sometimes it's made out of DNA and sometimes it's made out of RNA. We also need to know if the molecule is single-stranded or double-stranded, because we see both in viral particles. Next, it gets classified according to what's called its sense designation. This is something that relates to how the genetic material gets copied and whether it has a positive sense or a negative sense. This only refers to single-stranded pieces of genetic material. Next, it gets classified as to whether or not the virus contains an enzyme, this time called reverse transcriptase. You may have heard of reverse transcriptase because of its association with human immunodeficiency virus. This is a viral enzyme that is capable of doing transcription in reverse. Remember the central dogma. The central dogma tells us that transcription is the process where a DNA molecule is used to make RNA. So reverse transcriptase takes an RNA molecule and uses it to make a DNA molecule. That enzyme, reverse transcriptase, is not found in human cells or any other living cells. It's a viral enzyme, and it can do that strange process of reversed transcription. Finally, the Baltimore system is going to classify a virus based upon how the host cell replicates the viral genetic material. In other words, what is the name of the process that the host cell uses to make copies of the virus's genetic material? So 4 individual pieces of information that go into the classification using the Baltimore system. On this slide, you're looking at an image that shows us again some of the ways that viruses get classified using this system. For example, you can see numbers 1, 2, 3, all the way to 7 in this part of the drawing. Remember, one of the things that the Baltimore system classifies by is what the genetic material looks like, whether or not it's made out of DNA or RNA, and whether it's made out of single-stranded or double-stranded nucleic acid. So, a virus is going to get this 1 designation if it contains double-stranded DNA. It's going to get a 2 if it contains single-stranded DNA. A 3 for double-stranded RNA, a 4 for single-stranded RNA, and this one has a plus next to it. That's the sense. 5 is single-stranded RNA with a negative sense, and so on and so on. Remember, our cells, our living cells, all use double-stranded DNA, and they do a process called transcription to make RNA from that DNA. Our cells and all cells do the same thing. That's why we refer to it as the central dogma of biology. Viruses aren't living though. And viruses do something very different. They can do many different things because they have such variety of genetic material within them. So the Baltimore system is going to put a virus into one of these 7 categories. And again, it's not a naming system. It's just a way for us to talk about viruses and know a little bit about them according to which category they fall into.